Hey, good morning, y'all. Happy snow day, or not snow day, ice day. Um, I'm an idiot. I still went into school thinking we would start 2 hours late, but we aren't. It's an e-learning day. So what I wanted to do today was I want to take a ton of your time, but I want to go through the last little section of notes and prep for the quiz tomorrow. First off, I will say, yes, we still are having the quiz tomorrow. I'll go over these notes. These should be just review from what you guys had in your Cornell notes and the team activity, but I'm going to walk you through them, and then I will put a link up later that if you have email, if you want to have questions, you can come on. Maybe I'll make a Zoom link, or you can message me and pop on that Zoom if you have an individual question. Again, tomorrow I'll be in early like I always am, so if you have questions, you can pop on in. But this should go through and be pretty straightforward with what you're thinking, okay? So I'm going to zoom in on my screen here on my iPad, and we'll just walk through this last set of notes. So again, remember yesterday we were talking about just types of transport, and we specified osmosis. So we were very much looking at how osmosis, the movement of water, can create cells that will get really big or get really shriveled up or be normal sized. So make sure you understand the hyper versus hypo versus isotonic solutions. But today the focus was going to be on just general cell transport of the solutes. Yesterday was about osmosis, which is the solvent, the water moving. Today is more about the solutes, which, remember, is the dissolved particles, the dissolved substances. Okay, so we basically break it down into two types. Type 1 is what we call passive transport. Okay, passive transport is the movement of materials without the use of energy. Okay, no energy is required. This is what we call concentration driven. So again, you've seen this, but I drew this, right? And we said this would be the high concentration, down here would be the low, and this solute is going to roll down this hill free. No energy required. That is the example. Remember when I sprayed my spray in class, right? Oof, smells actually pretty good, but highly concentrated, low concentration, that is passive transport. Now there are variations of it, okay? So on the next slide, this is the most simple. This is called diffusion, right? And it goes directly through the phospholipid bilayer. These are small particles, things that are lipid soluble. They dissolve in lipids, so they can actually pass right through that membrane. Things like oxygen, things like carbon dioxide, things like very small lipids, things like alcohol. All of these will pass directly through the membrane. Again, a lot of this is really important stuff, oxygen, carbon dioxide, small stuff. Something like alcohol, the reason when people drink that it affects them so quickly is because it passes through this barrier very easily. All right, so that is what we call simple diffusion. Okay, that one's pretty easy. We also have another version of diffusion, but it has to go through one of these pathways. These pathways are those proteins, those channels. This is called facilitated diffusion. Remember, facilitate means to help. Sorry, I had a little hiccup there. I thought I wasn't recording. But the facilitated diffusion means you have to have help, and that's what goes through that protein channel. So this is also concentration driven. And examples of this, again, I put C right here because we already talked about this one. Osmosis with water. That is an example of facilitated. And if you remember, water uses a specific protein called aquaporin, right? And that was one that we mentioned the other day. But I wanted to make sure. So if this was a water molecule, that right here would be an aquaporin. It fits through, but that's all that fits through. Could be things like ions. It could be things like salts, things that have a charge. It could be things like glucose. Could be things like large proteins, right? Big things going through, or things that have charges. Those are the types of things that can't just enter through the wall itself. They have to have help, but they still go from high to low because it is still diffusion. So that is the second one. And then the third one was osmosis, which is the movement of water. Same deal. So those are all passive transport. If you look at this one, it's just a quick look of how it does it. This right here would be a carrier channel. If you fit, you go through. There's no door, there's no gate, you just go right through. This is how water goes through. This would be aquaporin. This one, on the other hand, you see how it has to go in, lock in a specific shape, and then once it's in, it then opens and allows it through. This would be a carrier protein, but this would be what we term a gated channel. In other words, you sort of have to have the key to fit, and then the door will open so that you can come through. But again, we are still going. You can see this is still high concentration, and this is still low. Still passive, still for free, but the gate has to be open. So that's how it works. That is passive transport in a nutshell. And again, this is just kind of highlighting all three examples. All right? First thing you should look at when you look at these and try to figure out what it is, you look at the concentration. There are more up here than there are in here. Then it would be high to low. So all of these are diffusion. They're all passive transport. This one is simple diffusion because it goes straight through. So this is simple diffusion. This one is going straight through as well. Small lipid insoluble solutes, so it has to go through. So this is facilitated diffusion. And then this one is going from high to low through that protein as well. So again, it is facilitated diffusion. But this one is through a gated channel. And this one is just through a simple channel. So that is all passive. High to low, either straight through diffusion or facilitated diffusion. So that's one. The other version that requires energy is called active transport, right? And all we really need to say right now about active is that it requires energy. Remember that energy comes in the form of ATP, that's our energy, our currency, and goes against the concentration gradient. So right here you can see these are examples, right? Here is a high concentration of ions, a lot of dots down here, not as many up here, but these are getting forced against the gradient. So energy is required to actually push them through. And again, if we did this, here's high, here's low. Right now we're taking that and we're pushing it up the hill. So literally there's energy and it's pushing it up the hill. That's what active transport is. The classic example is something like this. There's another example right here. It's called a sodium-potassium pump. And in this case, energy drives the transport. This one's kind of funky. I'm going to zoom in. This one is a 2-for-3 exchange. These NAs, 3 of them, will go in and load up this side. ATP, or energy, has to be inserted in here. And then the phosphates are going to come through one way. So it's sort of a revolving door, if you will. The potassiums will come in. That opens them. They go out. Then the sodium go in. Energy then pushes them back out. So it's called a pump because it requires energy use to make this thing work. So again, if you start, it's a cycle, right? So let's start up on the top right here. 2 potassiums go in. They come through, right? There they are coming out. Both spots are empty. Sodium reloads this side. ATP, or energy, forces them through. They go out. Now it's ready for potassium to come in again. This is the logistics of why you have nerve impulses. Sodium-potassium pump creates what's called an action potential in your nerves. So this action, we'll get more into it later, but as the base level, it is a type of active transport because it requires that energy. Again, just a quick reminder, ATP, that is the energy molecule. If you look at what I have right here, the ATP, the adenosine, right? This part is here. The triphosphate is here. There's 1, 2, 3, and then that red is just a sugar. But this is the energy molecule. And it's kind of cool, the way we used to show this and make sense of it in class. Notice I have 3 markers, 3, 3, 3, 1, 2, 3. These are the phosphates, right? I'm the sugar and the adenine part. Basically, what I have right here is an energy molecule that is full. It's like a charged battery. If you needed energy, I would come over, like I'm guessing you do right now, and I would pop off one of these phosphates. I would go, took that one off, but I would do that. That releases the energy. Now this is a uncharged battery, which through cellular respiration later can get recharged and ready to use again. Pop off, give energy. This is ADP, diphosphate, means 2. Can add this later. Now I'm fully charged. Break it again. So that energy is what drives active transport. Okay? Last thing I'm going to mention is right here. I got 2 slides left, one that you have, one that you don't. But it isn't always small molecules or smaller molecules. Sometimes it's big groups of stuff. So we have this idea, and I'll highlight it, and it's basically happening right here, right? Moving large materials or large groups of stuff out is called exocytosis. And again, I'm all about the power of words. Exo means out or exit. Cyto means cell. So stuff is moving out of the cell, okay? How does it happen? Well, here, let's say that this is a lysosome. Lysosome destroys all those old cell parts, puts them into this big old vacuole. That vacuole will slide over to the edge of the membrane and basically bond into it and then empty itself. So realistically, that cell just got a tiny bit bigger because this bonded with the outside and ejected all of that stuff. That is exocytosis. So the lysosome made a vacuole of all this garbage. It floated over. It bonded with the membrane and kicked it out. That's exo. I'm guessing you can figure out what the next one is. Moving large materials in. Well, the word for in is endocytosis. Now, endocytosis is the exact opposite. All right? You are taking a quantity of stuff. So again, right here, this is all the stuff. It's gathering right here on the membrane. The membrane then begins to pinch itself off. It pinches even more. And then finally, here's the membrane, and now we have pinched off a vesicle inside of it. This can then go to wherever it needs to go and deliver that components inside the cell. So this is called endocytosis. This happens all the time in your body. Exo means pushing it out. Endo means bringing it in. Realistically, when exocytosis happens, the cell gets a little bit bigger. When endo happens, the cell gets a little bit smaller. Makes sense. This one, and this is a new slide, I just wanted to show you there are 3 specific examples. And again, power of words. Endocytosis has 3 versions. There's one called phagocytosis, pinocytosis, and then there's a receptor-mediated endocytosis, okay? If you see the word phage, that means eat. So phagocytosis is bringing in food particles, actual physical particles. Pino means drink. So pinocytosis is bringing in extracellular fluid or liquids. So phagocytosis is bringing in food. Pinocytosis is bringing in drink. And then receptor-mediated endocytosis means I'm going to zoom in. On your membrane, you have these little receptors, okay? These proteins. This symbol, whatever that symbol is, locks onto here and locks onto here and locks onto here. And once those are received, the proteins create this little pit. That pit pinches in to become this. Okay? It's a way to not do this randomly. It's a way to only bring in certain things. Receptor-mediated endocytosis is how your immune system works, right? You have white blood cells. You have things that target specific things. Like if you've had an infection, if you had COVID, you've built up immunity to COVID, or at least some immunity, right? So, or I wouldn't say immunity. You've built up a resistance to it, a protection. So you have a receptor that knows what COVID looks like. And when it comes in here and gets in, it will latch onto it, and it will grab those COVID viruses, it will pull them in, and then this will destroy them. That's why we get vaccinations. What happens is you're introduced to a weak version of the flu. Your body makes these receptors so they recognize the flu, so that when you get it for real, they can recognize it much quicker. Can you still get sick? Yes. But is your sickness going to be shorter lived? Yep. Is your sickness going to be usually less? Yep. So, very specific. But that, my friends, are the notes. That's it. So if you have any questions, feel free to shoot me a message. I'm going to throw up a Zoom link. I think I'll throw it up here and be on the link for I don't know, maybe afternoon here. I'll have it open in case you have a question. You can pop on there. But have a good day off. Took a few minutes of your time here, but the notes are done. Quiz is tomorrow. See you tomorrow.